Hello everyone and welcome back for more biotechnology. So the time has finally come for us to really start talking about DNA in some good detail. So up until this point, especially based on some of the discussions that we had back in chapter 1, you should be pretty convinced by now that DNA is kind of at the center of the whole biotechnology universe. So we're definitely going to have a good time learning about it here in this chapter, and it's going to really be kind of the engine that drives a lot of our discussions moving forward. So let's go ahead and get into this. So like I said, we learned back in chapter 1 that most biotechnology applications are in some way going to manipulate DNA and other nucleic acids like RNA. Like I said, it's the engine that makes everything go. So we need to make sure that we do our due diligence in understanding what both DNA and RNA are all about. And obviously, they are both functioning as means of storing genetic information in the form of nucleotide sequences. So in this chapter, we are going to learn about various things like the molecular structure of DNA and how we can actually go about propagating it, isolating it from cells, and then actually going about and studying it and manipulating it. So in this picture that you're looking at, this picture is kind of funky looking. What you are seeing are DNA molecules that have been isolated from sperm cells from salmon, the fish salmon. So what you are seeing here is you've got this glass rod and you have this kind of thread-like sticky substance that is wound around the glass rod. So that is actually the isolated and purified DNA. So a technique called ethanol precipitation was used to cause the DNA to fall out of solution so that it can be wound around a glass rod like this. So in a later chapter, we will learn how this method actually works in getting DNA to come out of solution. So something that we did get familiar with, I believe in chapter 2, was something called the central dogma of molecular biology. So just as a little bit of review, this central dogma basically describes the way that information flows from DNA into messenger RNA. We call that process transcription. So a gene within a long DNA chromosome, the gene provides the instructions on how to make a single protein. With transcription, we make a messenger RNA copy of that one specific gene. And then with translation, a ribosome reads the messenger RNA and actively synthesizes a protein, the amino acids of which correspond to the codons that were found in that messenger RNA. So in humans, there are roughly 25,000 different genes that are spaced throughout the 23 unique chromosomes that we have. And we have 2 copies of every chromosome. So theoretically speaking, we should have 2 copies of each of those roughly 25,000 different genes. 1 copy from mom and 1 copy from dad. So, as I said, each gene is going to be responsible for the transcription or translation of one protein. So, 25,000 genes, that should be about 25,000 different proteins. There are actually quite a bit more different proteins other than just those 25,000 there, but we'll get into that in chapter 5 when we actually start talking about proteins, so you don't need to worry about it just yet. So something that we talked about back in chapter 2 was that DNA and RNA, both types of nucleic acid, are made out of nucleotides. So nucleotides have 3 different parts to them. The most important part is the nitrogenous base, and in this picture you can see 4 of the 5 possible nitrogenous bases. The ones that you're seeing here, cytosine, thymine, adenine, and guanine, are the 4 bases that you will find in DNA nucleotides. In RNA nucleotides, uracil will replace thymine. You don't find thymine in RNA, and you don't find uracil in DNA. So all DNA molecules, as we, I believe, have mentioned before, are going to organize themselves into that kind of telltale double helix structure where you have 2 different strands of nucleic acid that wind about each other kind of like a helical staircase. So each strand will have what's called a phosphodiester backbone. So in this picture, the backbone is signified by S for sugar, P for phosphate, and then that backbone just repeats sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate. The bond between the S and the P is called a phosphodiester bond. So this repeating backbone structure that you see here is called the phosphodiester backbone. And from this backbone, the bases are going to protrude. So the letters here indicate which of the 4 bases are protruding. So we have adenine, guanine, thymine, adenine, cytosine, guanine from top to bottom here. So the 2 strands are going to interact in such a way that the bases of each individual strand interact with the bases of the opposing strand. So you can kind of see that here. We have 1 strand on the left, 1 strand on the right, and you can see the bases are pointed at each other from each of those 2 strands. So which types of bases like to interact with other bases is not random. This is actually predetermined, and we call these rules for base pairing Chargaff's rules, based on Edwin Chargaff, who actually figured all of this out. So Chargaff's rules say this: adenine will always pair with thymine, or uracil if you're talking about RNA. So A should always go with T, or A should always go with U, depending on whether you're talking about DNA or RNA. And then likewise, guanine will always pair. As the rule, G does not go with A, C does not go with T. It's always A and T, C and G. A and U, C and G. Those are the rules, and we have to stick to it. So because those are the rules, the amount of adenine in one strand will always equal the amount of thymine in the other strand. The amount of cytosine in one strand will always match the amount of guanine in the other strand. So some other physical properties to talk about here is that the 2 strands of complementary DNA are going to be anti-parallel to each other. So that's probably a term that you may not have heard of before. So I'll give you the benefit of the doubt and assume that you know what parallel means, right? So when 2 lines line up right next to each other, they are parallel to each other. So what the heck does anti-parallel mean? Well, 2 lines being parallel to each other doesn't give any sense of direction. Anti-parallel assumes that the 2 lines are pointed in a particular direction, and DNA strands are no different. DNA strands have a directionality to them, and the direction we signify based on the location of the phosphate on one end and the sugar on another. The 5' end of the DNA is the end that has the free phosphate up here. So this would be the 5' end of this strand right here. And then you see you have a free sugar down here. That is called the 3' end. So the direction of this strand goes from 5' to 3', from top to bottom there. So when you look at the opposing strand, you can see that the directionality is reversed. You have the 5' end down here at the bottom and the 3' end up here at the top. So this is what we mean when we say anti-parallel. They are parallel, but the 2 strands are running in opposite directions. So that is what we mean by anti-parallel. So even when you are referring to or reading off the sequence of a DNA molecule, the convention says that you should always read it in the 5' to 3' direction. If you're having a little bit of trouble kind of picturing this in your mind's eye, picture 2 trains that are running on parallel grades of track. One train is going north to south and the other train is going south to north. They are parallel to each other, but those trains are moving in opposite directions. So that may help you kind of picture it in your head. And like I said, by convention, the sequence of a strand should always be written as 5' to 3', no matter what. And then the dots that you're seeing here that are in between the bases, you may be wondering what those are. So those are the hydrogen bonds that bind the 2 complementary strands together. So what you may notice here is that the adenine-thymine or adenine-uracil pair has 2 pairs of dots between them, whereas the cytosine-guanine pair has 3 pairs of dots between them. So each pair of dots signifies a hydrogen bond. So the adenine-thymine pair is held together by 2 hydrogen bonds. The cytosine-guanine pair is held together by 3 hydrogen bonds. So if we use our heads here, which base pairs should be stronger, adenine-thymine or cytosine-guanine? Well, if you say cytosine-guanine, you are absolutely correct. 3 hydrogen bonds should be stronger than 2 hydrogen bonds. It should be 50% stronger. So therefore, a DNA molecule that has a lot of cytosine and guanine should be held together more tightly than a DNA molecule that has a bunch of adenine and thymine. So if we consider some more physical properties of DNA, you can actually see in these space-filling molecular structures here, you can actually see that winding double helical structure here. The bases end up stacking on top of each other. So the way that this kind of helical structure works, you have base pairs that kind of line up. It's kind of like we're stacking them on top of each other. And the bases stack in such a way that there are 3.4 angstroms between those bases vertically. So angstrom probably doesn't mean a whole lot to you. So that's the same. So 3.4 angstroms would be the same as saying 0.34 nanometers. So a nanometer is 1 times 10 to the minus 9th meters. An angstrom is 1 times 10 to the minus 10th meters. So that's what we're talking about there. It's not super duper important. Each complete turn of the DNA molecule, so as you wind around that helix one complete 360-degree turn, that turn should contain about 10 base pairs. So the reason why this is good, the reason why it's good that we can kind of predict these things is that it allows there to be some uniformity throughout the entire DNA molecule in terms of structure. So a lot of molecular biology processes would not be quite as efficient if there were little differences or fluctuations here and there throughout the DNA molecule. It allows there to be this uniformity so that different processes can happen the same no matter where on the DNA molecule that they are happening. Something that your book does not mention, at least as far as I've gotten in it, is the presence of what are called major and minor grooves in the DNA. So you can actually look at this picture and you can kind of tell what I'm talking about. So you can hopefully see my mouse here. So you see how I'm kind of pointing to this gap right here. So this little kind of slanted gap right here. That is called the major groove of the DNA. And then if we come down here, this gap right here is not nearly as wide. That is called the minor groove. So DNA, the way that it winds about itself, it exposes major grooves, then minor grooves, then major grooves, and then minor grooves. So these grooves are important because enzymes can come in here and kind of stick little portions of the protein structure in here and detect what base pairs are present. And that's going to be important when and if you get to, say, a cell biology class. Those enzymes can tell what base pairs are in there so that they can know, okay, is this the gene that I'm looking for? Is this the gene that I want to transcribe into messenger RNA? So that's just something that your book doesn't necessarily mention, but you might find it interesting there. Okay, so let's talk a little bit about DNA replication, something that we have not really talked about at all here. So we've mostly been focused on the part of the central dogma of molecular biology that talks about how proteins are made. But part of the central dogma does tell us how DNA is replicated when a cell goes through the cell cycle. So when a cell makes a copy of its chromosomes, we call that DNA replication. So if a human cell has 46 chromosomes, it will need to make copies of those 46 chromosomes prior to going through the cell cycle. So momentarily, it will have 92 chromosomes, and it will put 46 in one daughter cell and 46 in another daughter cell. So during this process, the double-stranded DNA is going to be peeled apart. So we're going to take those two strands and peel them apart from each other so that we now have single strands. And each one of those strands is going to act as a template for the replication of a new strand. And this is where Chargaff's rules really come to be important here. So the reason why a single strand of DNA can act as a template for building a new strand is because of those Chargaff's rules. A always binds with T. T always binds with A, C with G, G with C. So if we have just a single strand of DNA, we already know what strand is going to be complementary to that. So having just a single strand of template DNA is all you need to dictate what the complementary strand is going to be that you will be synthesizing. So this brand of replication is what is called semi-conservative. If it was totally conservative, we would be able to build a whole new strand of DNA from scratch. So it is semi-conservative in the sense that we maintain the previous strands of DNA, but we have to split them apart in order to make new DNA. So like I said, the sequence of nucleotides in the new DNA that we're going to be synthesizing, that's going to be dictated by what bases are present in the template strand. So let's get a little bit of practice with this. So here's the question. If the sequence of a DNA template is given in red here, what is going to be the sequence of the complementary strand that will be synthesized? So this template strand is read from 5' to 3' end by convention, like we mentioned. Hey, that rhymed, huh? So ignoring that, if we read this from 5' to 3', we get adenine, thymine, guanine, cytosine, cytosine, guanine, AACTG, CAATT, CCG. So that is our template strand that we have to work with. So if we were going to put together a complementary strand that we want to make, it might look like this. If we line it up perfectly so that everything matches up here, we get TAC, GGC, TT, GACG, TTAA, GGC. But the problem with this is that we have written this 3' to 5', and as we said, DNA sequences are always read 5' to 3', so we need to flip that around. So the complementary strand that we are going to synthesize here would read CGGAATTGCAGTTCGGCAT. So that answers our question. If our template strand is in red, our complementary strand that we are going to synthesize from scratch is going to be that one right there, using the red strand as our template. Okay, so now that we have a good idea of what kind of the structure of DNA looks like, that's not something that's ever going to really change from organism to organism. So the DNA of humans is going to have all the same physical properties and complementary base pairing rules as the DNA from bacteria. But there are some differences in DNA from organism to organism. The first and most notable is the number of chromosomes. So prokaryotes, like bacteria, they tend to just have one big chromosome and their whole genome is just on that one chromosome. They do have a number of much smaller plasmid DNA molecules, but that does not represent their whole genome. Those are just kind of accessory genes that they may or may not have depending on different conditions. Another thing that can vary from organism to organism is the size of those chromosomes, the size of the genome. So we judge DNA size based on the number of base pairs it contains. So the human genome contains approximately 3 billion base pairs. So all 23 of our chromosomes together add up to about 3 billion base pairs, while the E. coli genome is about 4.6 million base pairs. So the human genome contains almost 1,000 times more base pairs than does the E. coli genome. And that makes sense, right? We are much more sophisticated, much bigger organisms. We should have a bigger genome, right? Next is the number and types of genes. So we've talked about how the DNA contains genes, but it turns out that not 100% of the DNA is actually gene material. So not all stretches of DNA are actually what we would call coding regions, meaning a lot of our DNA actually doesn't contain any genes at all. So really what that tells us is that just looking at the number of base pairs, that doesn't necessarily tell you how many genes are going to be there, right? So some genes will contain, furthermore, some genes will contain just a few base pairs, quite a few base pairs because it codes for a very large protein. Every amino acid requires 3 base pairs, so if you're making a big protein, you need a big stretch of DNA in the gene. While other genes actually contain not very many base pairs because the protein it codes for is small. So these are all things to consider if you're trying to use the number of base pairs to be a launching point for predicting how many genes there are going to be. It's not really a very exact science in that regard. And then finally, the shape of the DNA. So prokaryotes tend to maintain their chromosomes as a big circle. So basically, there is no 5' or 3' end. Those two ends are ligated together, so you basically have a big circle of DNA. Whereas mammals, like us humans, we maintain our chromosomes in linear fashion, meaning we do have free 5' and 3' ends. They are basically just very big, very long, continuous strands of DNA that are basically just maintained as linear pieces of DNA. They are not circular in that regard. Okay, so that is going to do it for this first video on DNA. So go ahead and join us next time, and we will start talking about sources of DNA and how prokaryotic, eukaryotic, and viral DNA sources tend to differ from each other. So I hope you will join us then.